Welcome everyone, my name is Lynn and I'm a senior scientist with QIAGEN's Bioinformatics. And today I'm going to give you some tips and tricks for using IPA. Many of these come from different emails or phone calls that we've received from customers over the years. And my hope is that the information that I share with you today will be really helpful for you, help avoid some technical errors that could arise, and just make your entire experience much more smooth and easier so you can get the answers to your questions quickly and generate new hypotheses. So the agenda today, I'm going to start with how you would first look at IPA going to the application preferences. And as many of you know, you can use IPA with a data set and also without a data set. So I'm going to first start with the steps that you would take from the data upload all the way through looking at a core analysis, and then I'm going to talk to you about some of my pathways features which we can use with the data set, or you can also use it with the different analyses that you've generated. And then I'm going to tell you about some additional resources that you can use to help you understand how to use IPA better, and then if we have some time, we'll have some time for questions. Okay. So let's start with application preferences. Oh, and one thing I forgot, the numbers refer to the slide numbers, which is way down here at the corner, in case you're wondering where we are. So how do you find application preferences? It's really easy. You just need to go to the File, Preferences, Application Preferences. And when you do that, you'll get this new window that pops up. And a lot of the things that are in application preferences are things like changing the font size, changing the coloration. But how many, raise your hand if you have a browser and you like to have more than one tab open? Many of you, yeah. I love that. And so the nice thing is in IPA, you can also have more than one analysis open. You can have several pathways and networks all at the same time. So I have mine set at the max because I like all those things open. So you can go up to 10 for pathways or networks or 5 for different analyses. And I'm going to tell you about some ways that you can avoid some technical errors using application preferences. So this is an error message that sometimes technical support will receive. It's GC overhead limit exceeded. So what this means is that your computer just hasn't given you enough oomph for you to run some of the heavier applications like bioprofiler or doing comparison analyses. So there's a really easy fix for that in application preferences, and it's a system memory allocated to IPA. So you can go all the way up to 8,000 MB. So anywhere that's greater than 100 or 1,000, excuse me, should be helpful for you. And I'm going to tell you what one more technical error thing that you can work with, and that has to do with clearing your cache. So sometimes what happens when we get a report from someone saying, oh, we have this error, and it's something that we haven't really seen very much, we'll test to see whether it's a bug. A bunch of different people will use a bunch of different platforms and try to figure out, can we replicate this? And if we can't, one of the things we'll say is, did you clear your cache? Which I'm sure many of you have used programs and you've had customer support people tell you that. And sometimes when I say that, I hear this little panic, like, oh my gosh, this is going to be so hard to find. But we make it really easy because we have a cache tab. And I kind of refer to this as the magic wand because sometimes it just magically gets rid of any issues due to these temporary files that may have been corrupted. So you can increase the size. By default, we have the cache size set to 200, and you can go ahead and go all the way up to 10 gigs if you like. And one important thing is any changes that you make to your application preferences, you do need to hit that save button that's down on the lower right, log out of the program, and then log back in because otherwise the changes won't take effect. So now let's start talking about how to use IPA, give you some tips and tricks for starting with the data upload. Okay. So one of the most common things that we hear from customers is, I have all these IDs, I uploaded my data set, and a whole bunch of them didn't map. So what do I do? Or I want to take my gene IDs and I want to have them as UniProt IDs or vice versa. So this BioDBNet is from the National Cancer Institute. It's a free web-based software, you can go here and you can just copy and paste your IDs in here and just automatically convert them and then upload that information into IPA. Another thing they have is this, something called DB Ortho. So as many of you know, IPA is limited in its findings to human, mouse, and rat species. But you can use other organisms for IPA as long as you have gene IDs that are orthologs to human, mouse, or rat genes. So a very easy way of finding it is to go to the DB Ortho section, and here you can just put in your gene IDs and put an output, okay, what kind of organism do I have? So here I have the little brown bat, and I want to convert those to human IDs, and then I can just submit that really fast, get that list, upload it into IPA. With metabolomics IDs, one of the newest things that has happened that we tend to get a lot more people who are asking metabolomics related questions. They'll contact us and say, you know, I uploaded my data set and you're not recognizing any of my HMDB IDs, so what's wrong with you all, right? And so it's like, well, actually, a lot of the software that's using metabolomics information has the older version of the identifiers for HMDB. So those are 5 digits. And we're accessing the latest version of HMDB, which is 7 digits. And it's actually pretty easy to make that conversion yourself. All they did was added 2 extra zeros at the beginning. So you can just go through maybe an Excel or text editor and add that in, and IPA should be able to recognize them. And as I've mentioned here on the slide, you can see that we have 4 different types of identifiers that we'll recognize in IPA. And a lot of times, the data that I've seen will have someone who'll say, I have a bunch of HMDB identifiers, my core facility gave it to me, but you know, there's a whole bunch that I just have the names for and I don't have any other identifiers. So is there a way that I can easily find more identifiers so I can use IPA? And there's a bunch of different websites out there. I think my favorite is Embrol. And so you can just put your IDs in, make selections, and hit submit, and you can get a nice, maybe some of them are going to have HMDBs, but maybe some of them only have PubChem information. So this is one way that you can help maximize the number of genes or proteins or metabolites that you'd like to analyze. And another thing that we did to improve mapping is in March, the release, we introduced the ability to have multiple ID columns. And so this can really help maximize the percentage of molecules that are mapping. So you can see here that out of 409 identifiers, that only 307 mapped. But if I also include my other identifiers, I get up to 379. And this can also be really helpful too in situations where I've seen cases where some PubChem IDs will actually map as genes because the 2 databases are, you know, IPA saying, oh, well, that looks like a gene identifier, but it's not because there's this crosstalk between the 2 databases. So this is something that you can use to help solve that problem. In terms of microRNAs, sometimes people will just upload their list of microRNAs, run a core analysis, and then say, well, how come I'm not getting any pathways? All my networks are just microRNAs. And so the reason for that is that we need to have the mRNA target, the ones that are targeted by the microRNAs. So what you can do is go and use our microRNA target filter and very easily upload your data set, find the mRNAs, then upload those into IPA, run a core analysis. But you're not going to lose information by not having those microRNAs because you can later add them into whatever pathway or network that you find really interesting. And importantly, if you're going to use the target filter, you're going to want to make certain that you map them as the mature version and not the stem loop version. So you can do that there when you're setting up, telling it which identifier type to use. Another thing is sometimes people will contact us and say, you know, I have this list of microRNAs, and most of them are not mapping. And the ones that do are wrong. There's something wrong with your software. Can you check in on this? Is this a bug? And as those of you who have worked with microRNAs will probably realize historically the nomenclature was kind of like the wild, wild west. And it's only been recently that we've had consensus as to what microRNA should be. So if the software that you're using is looking at like Target Scan version 17, but we're accessing version 21, there can be significant differences. The other reason is there might be differences is because what we do with the mature microRNAs is we do cluster them into entities. We're basing that on the 7-seed sequence similarity. And the reason that we do that is it does help with improving the specificity of that targeting information that we get from our different databases. Another thing that you can do is use what's called the batch upload feature. So if you have a whole bunch of different data sets that you want to upload, you can format that just like you would a regular file, but then at the top, you're going to put metadata on there. And so here's a really very simple example of that. And then what you can do is go into IPA, just select the batch upload feature, and then you'll get this window that pops up and it'll ask you, well, where are your files? What directory are they in? And where do you want them to be stored? Okay. And then another thing, I know that Stuart and I think Jean-Noel mentioned this process for using the search, for adding, and this is a really great thing to do when you're trying to use, say, the OmicsSoft repository. You want to overlay your data, run comparison analyses. And you can add that during the data upload process in this metadata tab. So here I'm showing you an example. There's a lot of different keys that you can use, different key terms. You can add your own key terms, but it does make it easier when, 5 years from now, you're still using IPA and you're like, okay, I did a data set on melanoma. Where did I put that? You can do that, but the nice thing too is that I did a search here for melanoma and I had 4 different samples in my files, but there's over 2,000 in that OmicsSoft repository. So there I can go ahead and take my data and make comparison analysis and overlay, and it's just a really nice, powerful process. Okay. All right, so moving on from data upload, now I'm going to tell you about some tips for creating the core analysis. Okay, so one thing that I suggest is enabling the causal network feature for those of you who have the advanced analytics license or higher. And not just because causal networks is a really cool thing to have, right? It's very interesting to see master upstream regulators and their networks, but this can have a big effect on your analysis match results. If you don't enable the causal networks for that analysis, your query analysis, you're not going to see anything in the causal network column in your analysis match results. There's nothing that you really can match those up with, okay? So to make your analyses and your matching much more powerful, I strongly suggest that you select that causal networks. And you can see here that once I did that, now I have a lot more information and I can make much more accurate matches. Right. Another question that comes up sometimes is people will have metabolomics data sets and they'll map it. And then they'll say, I don't have any molecules that are mapping. This is the message that I'm getting. And we have realized the problem is that they've unselected, uncategorized. So a lot of the metabolites we have in the knowledge base, we don't have information from where they're from, what species they're from. So if you deselect uncategorized, you're going to probably get a message that looks just like this. And another thing that you want to consider too is if you have something like the little brown bat, you aren't going to say, oh, that's only uncategorized. Uncategorized doesn't mean that it's a different species, just that the location or where the organism, where the findings came from, we don't, the finding didn't really indicate where it was from, species it was from. Okay, so that's what it would look like if I selected uncategorized. Okay. Another really important thing is considering your reference set when you're setting up your core analysis. So your reference set is the background set of molecules that you're going to test your most significantly perturbed molecules against. And that's going to generate the p-values that tell you which pathways, upstream regulators, diseases and functions, et cetera, are significantly enriched. So that reference set is essentially the molecules that you tested. So RNA-seq be essentially or theoretically the entire genome. But sometimes what happens is we have smaller data sets. And in that case, you didn't really test the whole genome, right? So let's imagine you had a QIAGEN RNA targeted panel on inflammation because you thought inflammation is really important for your research. So you tested 500 genes. And when you set up your analysis, let's say that you chose the Ingenuity knowledge base for your reference set. Do you think you're going to get amazing p-values for inflammation-related processes, or will they be what you would expect? What do you think? Great p-values or Amazing. You're going to amazing where you run down the hallway saying, these are the most amazing p-values I've ever seen in my life. Amazing. But guess what? No, because you started with just inflammation genes. So of course you're going to have amazing p-values for inflammation. Okay, so what you want to do with these small data sets, you're going to upload the whole thing, just like you normally would. And so these are the genes that are in this box are the ones that are significantly perturbed, in this case, highly dysregulated. So I'm going to set my cutoffs to find those genes, and I'm going to test them against everything in my data set. And the way you do that is in your general settings, you're going to put the reference set as your user data set. And then down below in this box, you'll set the cutoffs to determine which of these molecules are significantly perturbed, highly dysregulated, et cetera. Okay. And then finally, I'm just going to give you a couple little tricks that you can do when you're working with a core analysis. And the first one is, let's say you're looking at your upstream analysis results and you see that there's two upstream regulators and you'd love to really just quickly and simply put them all into the same network. So what you can do is really easy, just select both of them. In the table, go to Display as Network, and then your network will be generated, and you can see that both of those upstream regulators are now in there. There's ways that you can do it a different way in IPA, but this is one that's probably the most simple way of doing this. And finally, I want to tell you about a little trick for using generate networks and regulator effects. So I think Jean-Noel and Stuart mentioned regulator effects, the upstream regulators, data set genes, and the linked diseases and functions. But maybe you look at this and you go, well, that's great and everything, but I'm really not interested in organismal death. So can I, instead of just deleting it, can I regenerate this network? And the answer is, of course, yes. And the way to do that is you first need to find what overall category does this belong to, because we do have this hierarchy in our Ingenuity ontology. Organismal death is kind of at the bottom. So you can just simply go to the Diseases and Functions tab, which is right next to Regulator Effects, look down on the table. We see right here, it's an organismal survival. We'll keep that in mind as we go back to Regulator Effects. Click on the Generate Networks button, and then we can deselect organismal survival, hit OK. It will regenerate the networks without that particular organismal death. And you can also do things like change the p-value cutoffs, z-scores, change which molecules you want to consider as well. I think this is an underutilized function, and I just wanted to point that out to you all today. So now moving on to the My Pathways tools, which we can use if you just have a question that you want to ask IPA, but you don't have data, you can use these tools. And of course, if you find different pathways or networks that you find really interesting, you can also use these tools on them. So the first thing I want to talk about is the Connect tool. So this allows you to take different nodes and show the relationships among them graphically. And you do need to select molecules that are on the canvas to even be able to hit the Apply button. The great thing about the Connect tool is that if you select, you can have as many connections and many molecules that you connect as you like. And let's say in this example, we want to take this drug right here, and we want to know what are all the molecules in this pathway that could be affected by this drug. If we selected all of the molecules on the pathway and we hit Connect, this is what we would probably see, or this is what I saw when I took the screenshot anyway, and I just went, oh, right. This might be something that you're really interested into, and this should be the only time you hear it in a talk today or tomorrow, by the way. But instead, if you just want to know, you can just select the drug and hit Apply. And boom, we only have 2 molecules on this pathway. Okay. All right, another way that you can find connections between nodes that are on pathways or different networks is to use Path Explorer. So here you take maybe molecules or maybe a disease, and you can put those nodes into one set or another set, and you can ask, can we find any connections between these molecules or these diseases? So here, when we hit the Apply button, you can see there's a table here beneath that canvas where we can see that there's intermediate nodes in between CASP12 and MCL1. And we can actually add that straight to the canvas that way. So we can find connections that may have been hidden this way. Okay. Another thing that you can do is, let's say that you're interested in growing. So using the Grow tool allows you to find molecules that are maybe upstream of a molecule of interest or downstream, or both. And so what you can do here is, let's say that you're interested in CDC27, but you want to know not just what's in the knowledge base of molecules that are affecting it or are affected by it. What about just my data set? That's all I'm really interested in. So what you can do is you can filter to your data set. When you select this button, you'll get this window that appears. It'll take you to your project manager, and then it will just grow to what is in your data set. So this is color-coded just like you would see in an analysis. So green is downregulated, pink, red, upregulated, and the other molecules, because I chose an analysis and I had set cutoffs, those green ones, or the gray ones, excuse me, are in my data set, but they were not considered to be significantly perturbed from my cutoffs. But we've included them there too. And another thing you can do is microRNAs. So let's say you want to do the exact same thing, but you want to find out what are the genes in my data set that could be affected by this microRNA. So just like I showed you before, you can select your data set, and let's say that you want to get really strict. Maybe there's a whole bunch of different genes, and you just want to narrow it down to those that have been experimentally observed from the literature, rather than those that have been predicted to be targets. So here you can use this confidence level filter, and you can just select experimentally observed, and then we get just a couple of genes in the data set. It helps you narrow down onto what you consider to be most interesting. Okay, so right now I hope that the tips and tricks that I've provided for you today are very helpful. I don't want to just walk away. You know, it would take days and days and days for me to show you every single potential tip and trick in IPA, as many of you have experienced. It's a little complicated, but a very exciting program. We do have a lot of recorded webinars and video tutorials that are available to you to view. It's at tv.qiagenbioinformatics.com, and we have a whole bunch of different topics. And the other thing I want to let you know too is that we do have an online help manual. If you weren't aware of that, you can go to that website, and this is a website you can actually access when you're not an IPA. So let's say you're at an institution and you have like 10 seats and you have to sign up for it. You don't need to be in IPA to access the online help manual with that link. But if you're in IPA, you can go to help and support, and it will take you straight to that same link in your browser. And we have a lot of different articles, so if you're the sort of person who doesn't want to watch a video, you read faster than you can hear, that's a great resource for you. And of course, we have a great technical support team. We have people who are ready to answer any question you have. You can call us. Our email is the TS, which stands for Technical Support Bioinformatics at QIAGEN.com. We do promise to respond within 1 business day. That is the goal, and we always meet that goal, always. And oftentimes you get a response within a few hours or later that day. And we're open for most hours of the day. And then, of course, here are our other websites.